Thank you. So my name is Chris Smallwood from the University of Tennessee. I am working with the National Association of Plant Breeders to host the 2017 NAPB webinar series called Building Better Plants for the 21st Century, Updates and Upgrades in HTP, IP, and DNA. We hope that you were able to join us 2 weeks ago when we heard from Russell Jessup speaking about IP learning curve on the job or graduate curricula. We're glad that each of you can join us today, where we are hearing from Phil Simon, USDA ARS in Wisconsin, speaking about building better carrots for growers and consumers. And then in 2 weeks, we hope you'll join us for Jim Myers from Oregon State University when he discusses trends in intellectual property protection in plant breeding. And with that, I'm going to turn it over to Adam for a more in-depth introduction.
Thanks, Chris. Hello, everyone. My name is Adam Bolton, and I will be introducing today's speaker and my own academic advisor, Dr. Phil Simon from the Department of Horticulture and the USDA ARS at the University of Wisconsin-Madison. Dr. Simon's background is in carrot and garlic genetics with a focus on improvement and release of widely used carrot germplasm with nematode resistance, higher nutritional quality, increased flavor, and novel purple color. Dr. Simon was also responsible for leading the initiative to publish the carrot genome just last year. Today, Dr. Simon will be speaking about building a better carrot for growers and consumers. So thank you for joining us today, and I will now switch over control to Dr. Simon.
Thank you, Adam. I appreciate the introduction, also to you, Chris, and to Mark.
Okay. And we've got a quick poll we're going to launch here. So I'm going to do, for our attendees, you should be seeing a poll come up. It's just a couple of quick questions. We're just going to take a real quick, just about 10 seconds, and this will allow us to know a little bit more about your background and maybe your exposure to having grown carrots or produced seed via carrot. And just another 5 seconds. Okay, and let's go ahead and end that now. And then, Dr. Simon, you may not be able to see these. Are you able to see the results there?
I am.
Okay.
Yeah, I can see them. Yep.
Okay.
Thank you.
All right, I'll go ahead and stop that now.
All right. Are we ready to go?
Yep.
All right. Well, thank you. I appreciate your input on the poll. I see that most of you have grown carrots, but at least as a root crop, but have not grown them as a seed crop. And I'm not too surprised because it's not a crop that commonly is handled as a seed crop. Today I'm going to talk about what I'm entitled Building a Better Carrot for Growers and Consumers. And this refers to, let's see, there we go. This refers to the domestication and modern breeding of carrots. I'm going to break my talk up into two different parts. The first part will be a brief history of carrot domestication and that initial building process of the crop that we know today as carrots. So let's start actually before carrot domestication in prehistory, at least as prehistory as carrot goes. Wild carrots thought to have occurred in Asia and Europe for a long time. The scientific name is Docus crota, subspecies crota. The first evidence for human interaction with carrots comes from the observation of carrot seed found at ancient websites 3,500 to 5,500 years ago in Germany and Switzerland. This is, again, the first evidence for human use. Likely this carrot seed that occurred at these campsites was not seed for a root crop, but rather was wild carrot seed likely collected and used as a spice or medicine, like the other Apiaceae. And we see along the bottom of this slide some images of not only carrot seed, but some of its relatives, caraway, fennel, and coriander, some very aromatic and spice seeds. And again, these likely were seeds not from a cultivated carrot crop, but rather from wild carrots. Let's see here. There we go. So what these wild carrot plants looked like 3,500, 5,500 years ago is likely what carrots look like today, or at least wild carrots look like today, which is actually what cultivated carrot flowering carrots look like as well. You see in this image a field of wild carrot in a vacant lot in Wisconsin. Below that, a vacant lot in Tunisia. And these are both natural production of wild carrots in these vacant lots, as carrot has commonly been in history. You see the flowering plants early stage above and in late flowering below. These wild carrot plants, from a flowering standpoint, look like cultivated carrots, but from the standpoint of the root crop are quite different. You can see that root crop on the right with white, very forked roots and a few plants flowering in them as well. Wild carrots, sometimes called Queen Anne's lace, and the reason for that is shown on the bottom of this right-hand side of this slide, with a picture of some lace and a carrot umbel, which to some eyes looks like that lace. So consequently, the name Queen Anne's lace. So as I mentioned, wild carrot is thought to have initially occurred only in Europe and Asia, and throughout these regions, and that's what we have here is Europe, Asia, and North Africa. And in fact, most of the wild Daucus species, that genus that carrots in, occur around the Mediterranean basin. There's roughly 20 Daucus species. A couple of them are crossable with carrot. Now, in contrast to where the origin of these Daucus species occurs, the origin of domestication of carrots is thought to be in Central Asia. That's the orange circle on this slide. The modern information confirming and Central Asian origin of cultivated carrot comes from some work we did a few years ago, displayed in this phylogenetic analysis in this slide. What we did was some SNP analysis in some cultivated carrots shown in blue on the right of this slide, and some wild carrots shown in green on the left of this slide. The point of interest for this slide in particular is on the bottom, or right at the 7 o'clock zone of that slide, central bottom, where you see wild C Asia, which is wild Central Asian carrots. Those Central Asian carrots, wild Central Asian carrots in this analysis were most closely related to cultivated carrots, and that's borne out in this phylogenetic tree. So this analysis involved an evaluation of 88 wild and cultivated carrots from broad geographic regions, and the bottom line from this study is that the wild carrots from Central Asia were genetically most similar to those cultivated carrots, suggesting a Central Asian origin of domesticated carrots. A number of SNPs, some standard analyses. One point worth noting is there's been minimal loss of diversity with the domestication of the carrot crop. It's a very diverse crop, both in the wild and in the cultivated form. This study does agree with speculations by the body of historical information that carrot originated as a root crop in Central Asia about 1,100 years ago. And this is what brought Vavilov to suggest the Central Asian origin of carrots. Although there's some indication carrot may have occurred as a root crop in the Roman Empire, but it's not well supported. So moving to Central Asia today, we find many shepherds moving their crops around, as they likely were doing when carrot was first domesticated. And in addition to shepherds in this part of the world, we see, this is a shot of some shepherds in Uzbekistan. In some carrot collecting I did in Uzbekistan, there are also modern carrot domesticators or breeders of carrots. For example, this woman shown in the picture from Uzbekistan, who was growing some yellow and some orange carrots. And so the domestication process of carrots started with that wild carrot you see as a white, highly divided root, where early domesticators brought carrot into cultivation along with the process of selecting for smoother and yellow and purple roots, not white roots. So several of the obvious changes in the phenotype of carrot with the domestication process by these Central Asian carrot domesticators, and for that matter, Central Asian breeders today. So we're continuing this process that is estimated to have begun about 1,100 years ago, in particular in Afghanistan. I haven't been to Afghanistan, but came pretty close in Uzbekistan. And perhaps contrary to what you might expect, the first carrots were not orange, but rather the first cultivated carrots of the root crop were yellow and purple. Again, there's some evidence carrot might have been the Roman Empire, but the better evidence is that it started in Central Asia. From Central Asia, carrot as a root crop spread to the west through North Africa, Europe, and throughout Europe eventually, and to the east through India and China. Along the way, as I noted before, color was selected, yellow and purple initially, smooth roots, as I mentioned before, and also a tendency to not flower, because a flowering carrot is a carrot that is not edible. If you've grown carrots that have flowered, you'd know that. So those are some of the traits, but likely flavor was also selected because while wild carrot is edible, the flavor is very carroty and very strong. Now, the origin of orange carrots likely began in southern Europe in the 14 to 1500s, and that marks the last pre-modern change in carrots, with the orange types showing up in the 14 to 1500s in southern Europe. As I talk about breeding in a few minutes, we'll talk about the development of carrots and hybrid carrots in the 1950s. But just a quick review then, we started with this wild carrot the ancient domesticators started with this wild carrot on the left, and then selected carrots of different colors, initially purple and orange, but also red and, purple and yellow, and eventually orange and red carrots shown here as well. And this initially was done in Europe. So the written history of carrot indicates that it was selected initially as a root crop in Central Asia, but it's artwork that gives us the best indication of where orange color was first selected, where and when orange color was first selected in carrot. And we see here several examples of artworks from Italy, Spain, and France, where some earlier images, and one of the earliest of orange carrots depicted is on the left, this image by Sanzio. You may not be focusing so much on the carrots, which are actually embedded in the lower part of this collection of vegetables. You see an orange carrot in the lower left-hand corner in this early 1500s painting. So the first evidence for orange carrot shows up in southern Europe, places like Italy and Spain, in the 1500s to 1600s. And after that, orange carrots became the most popular carrot and continue to be the most popular color of carrots today. But along with this process of domestication and cultivation and breeding of carrots, we have a wide array of shapes of cultivated carrots shown here on the left. And in fact, modern carrot cultivars are classified, especially orange cultivars, are classified by their root shapes, and there's a large collection of them shown here on the right. So let's take a quick look at where we're at in the process of domestication and then breeding carrots today. So the global carrot crop today, most of the crop is orange. Some of these novel colors, at least what we think of as novel colors, continue to be grown, but most carrots grown around the world today are orange. If you're growing and consuming carrots in Asia, you're likely growing a Kuroda type with the shape of that carrot on the left of this image. And the Nantes type is the predominant shape of the carrot root grown in almost all of the rest of the world. So Kuroda and Nantes, the two predominant shapes. An exception to that is in North America, where we typically grow the Imperator type, which is the long, thin, more Bugs Bunny pointed carrot, shown here in the lower right of this picture. Another global trend today is that there is an upward trend in fresh carrots worldwide today. Some more facts about the current carrot crop. It is a widely grown crop. It's an interesting crop because some of the same cultivars can be grown north of the Arctic Circle in the summertime as what's grown in the wintertime at the equator. Relatively little photoperiod sensitivity. The value of the primary crop is stable or rising. It's interesting to note that organic production in the U.S. is high, accounting for about 14% of the U.S. carrot crop, and it is rising. There are some new high-value products of carrots. Those of you that are less than 25 years old probably don't realize that at one time, baby carrots, shown here to the right of this picture, were not available. It's a product that's only been around for about 20 years, which are pieces of long whole carrots peeled for the consumer's convenience. The seed value of the carrot crop is relatively low, and what that means is that it hasn't had quite as much interest from seed companies. Like other crops, there are new pest challenges, but in addition, there is greater interest in an adaptation of carrots to warmer and drier climates, because typically it's been characterized as a cool season crop. So that's my overview of the domestication process and early history of carrots. And maybe we'll stop at this point and see if you have any questions about some of this introductory information on carrots.
Okay, there's no questions at this time, but I would encourage folks to put questions in Q&A, and there's a quick poll we're going to launch just to maybe test your knowledge here. So we'll take about 10 seconds and let folks weigh in on in our poll, see if you were paying attention or not. And we'll take another 10 seconds. And okay, we'll go ahead and end that poll, and then I'll share the results there. And Phil, if you can see,
I
those,
can.
did folks pass?
Yes, yeah. Well, I specifically did mention that wild carrots are not poisonous, and people got that right. And they don't necessarily taste well, but they are not poisonous typically. I didn't mention it specifically, but carrots are among the more recently domesticated crops. Only about 1,100 years of recorded history of carrot as a root crop, as compared to some early crops that go back 10, 15,000 years for something like wheat and barley. So appreciate the feedback on that poll. Thank you.
Okay.
So moving on then, let's talk about modern carrot breeding. Some summary statements at the top. Carrot breeding is a relatively slow process. There are certainly other crops that are slower. And there are relatively few carrot breeding programs today. I'll talk about that as we go along here. First, on the topic of numbers of carrot breeding programs, in the 1960s was really the heyday in terms of the number of carrot breeding programs nationally, with 8 programs in Europe and Asia, public sector programs, about 12 public sector programs at land-grant universities, and the USDA in North America in the 1960s, and about 25 US seed companies, most of them being regional and national. Along the way, the consolidation of seed companies, plus the de-emphasis of some of the breeding efforts at land-grant universities, has resulted in a reduction in the number of national programs looking at carrot breeding and crop improvement, translating to about 3 in Europe. 2 public sector programs remaining in North America. Irwin Goldman's at the University of Wisconsin, and mine also at the University of Wisconsin. Irwin focuses on processing, canning and freezing carrots, I on fresh market carrots. A few public sector programs in the rest of the world, and about a dozen seed companies account for most of the supply of carrot seed globally today. These are pretty much all multinational, and the main focus is on breeding carrots as a hybrid crop. Some basic carrot breeding for those in the audience. This is a plant breeding seminar. Carrot is a diploid outcrossing insect-pollinated species, 9 pairs of chromosomes, relatively small genome, about the same size as rice. No self-incompatibility in carrots, but there is quite a bit of inbreeding depression. But like other outcrossing crops, we select for reduction of inbreeding depression in the process of developing inbred carrots. It is a highly diverse crop, both wild and cultivated. I had mentioned that earlier. There's relatively little reduction of genetic diversity, allelic diversity, when you look at cultivated carrots compared to wild carrots, suggesting perhaps multiple domestications or introgression of wild carrot as the crop has been grown. But whatever the reasons, there's significant genetic diversity in cultivated carrots as well as wild. Carrots referred to as a biennial, but it's easily handled as an annual in breeding programs, where we typically will harvest the root crop and vernalize those roots for 4 to 6 weeks, after which time they will start producing seed stalks, and I'll show you that in a few slides. From the standpoint of where we're at in terms of basic genetic knowledge of carrots, only about 20 carrots were known in 1985, whereas Now, today, there are roughly 100 genes and QTLs have been well characterized in carrots. About 15 traits have been reported for their heritability in carrots, and as was mentioned earlier, the carrot genome has been sequenced by our lab in the last year. This gives us abundant access to many markers, hundreds or of thousands or really millions of markers, which we're typically characterizing using genotyping by sequencing and other genotyping techniques. And with this process, we estimate we could save somewhere between 3 to 10 years in the breeding process as we're moving along. I'll point out here that hand pollinations of carrots are very difficult. Now, I'll come back to that in a little more detail later, but keep that in mind as we go along. So the process of carrot breeding largely involves phenotypic recurrent selection. All major new cultivars grown in the US, Europe, and Japan for large-scale production are hybrids today. But there still is some effort in the development of open pollinated carrots. And in fact, we have some of this work underway in my breeding program, in particular targeting them for some of the organic industry. But the large-scale production of carrots is with hybrids. Typically in the breeding process, we'll self-pollinate for the crop for roughly 4 to 6 generations after the F2 generation. At around F3s, we start back crossing into sterile cytoplasm. It's a cytoplasmic male sterile system in carrot I'll talk about in a minute. And then start evaluating combining ability for the production of hybrids at the F4, F5 level. So relatively straightforward from some other outcrossing crops grown as hybrids. So typically carrot trialing initially involves small trials, typically something like a 1 or 2 meter trial shown in the upper left-hand corner here with a group of people evaluating them in a sort of a beauty contest type of evaluation. For larger scale trialing at the grower level, typically these large scale growers will require at least 2/10 of a hectare of carrots to get yield data with their large scale harvesting equipment shown in the upper right hand corner. So the two major carrot types grown by large-scale growers in the U.S. today are the Imperator type sold as whole carrots, which is shown in the right on the bottom of the slide. So these would be carrots that would be washed up and put as whole carrots in the plastic bags. And also carrots grown for the baby carrot production, the cut and peel carrots that I showed you a picture of earlier, where these long, thin carrots are topped and tipped, washed up and cut into chunks and then peeled lightly to make those baby carrots you probably are familiar with. So after the carrot roots are harvested in the breeding process, during the vernalization process, we have this 4 to 6 week period of vernalization, which is done at just a couple degrees above freezing in a typical refrigerator. We have the process of some visual selection, selecting for something like the shape of the crown of the carrot, shown in the upper left, the shape of the tip, and an evaluation of internal characteristics like looking at greening in the shoulders of carrots in the center picture, and the elimination of carrots with things like yellow cores shown in the bottom of this picture. So at the end of this selection process, we end up with pieces of carrots where the bottom part of the carrot is removed, like this purple carrot shown in this picture, but with a centimeter or 2 of the petioles left intact. And just note in passing, this is in the lower right is an image demonstrating hybrid vigor in carrots with 2 different inbreds on either side of their F1 hybrid in this image. So going back to the selection process, during the vernalization, we'll do this visual selection. We'll also use this time to, as we remove the bottom of the carrot, to look at the internal color, to do some flavor evaluation, and to take samples for things like root composition for pigments, for instance. After we vernalize those carrots, these pieces of carrots, the bottom half of the carrot root with some of the petiole base attached to it, are ready for planting for seed production. This is referred to as a steckling, and these vernalized top half of the roots with that petiole base are then in the wintertime, in Wisconsin at least, planted in pots in the greenhouse shown in the upper left here, and or planted in the field in the image to the right with individual carrots in these white fabric cages and large numbers of carrots for hybrid production in the larger cages. In the middle is an image of some cages we use for insect pollination of individual carrots in the greenhouse or in the field. So that's the process by which we grow and trial and select carrots. A little bit about the carrot seed production and hybrids. So on the left here is an image of a carrot inflorescence with many hundreds of flowers, where each individual flower is quite small and very difficult to hand pollinate. In the middle is some drawings of some carrot flowers and cross sections in the lower right of this picture, some cross sections of carrot seeds. The typical carrot phenotype found in wild carrots is that shown in part A of this diagram, where you have 5 petals and 5 anthers and then the pistil in the middle. The other interesting phenotype that's the basis for hybrid carrots is shown in image C in this drawing, where the 5 anthers are replaced by 5 petals. This is referred to as petaloidy, and it's controlled by, it's a cytoplasmic male sterility, like all male sterilities reported in plants, controlled by the mitochondrial genome. So with the replacement of these anthers by 5 petals, of course, that plant then is male sterile, but female fertility is retained. So it's worth noting that the diversity of wild carrots played an important role in the identification of carrots for the development of the hybrid carrot, because it's male sterility, cytoplasmic male sterility, that's used for production of most carrot hybrids today, came from North American wild carrots, initially discovered by Henry Munger in Cornell in 1953, and another source discovered by C.E. Peterson and Teddy Morlock in the mid-70s in Wisconsin. A couple of people that were instrumental in the development of hybrid carrots were C.E. Peterson, who was my boss when I was hired, and Warren Buck Goblin. Both were at the University of Wisconsin when I met them, and they used that petaloid male sterility to develop the initial hybrid carrots that are grown today. And this sterility is used for nearly all the large-scale U.S. carrot hybrid production today. And we have some molecular markers to track that trait as well. And so this shows some large-scale production of commercial carrot seed production fields, similar to that wild carrot field I showed you earlier on in my presentation. And some pictures of some beehives here in the lower, in the right-hand image, because bees are typically used to pollinate carrots in the large-scale production, which is typically in eastern Washington, eastern Oregon, and Idaho. Some breeding goals for carrots today. Let's briefly go through them in a general way and talk about a couple in particular breeding goals, both for growers and for consumers. So from a grower standpoint, uniform appearance and size is important as it's machine harvested, and that's the impetus for the development of hybrids. Freedom from diseases and defects is important, of course, as well, and there are several major diseases. From a consumer standpoint, flavor is very important. Sweetness, lack of turpentine, harsh flavor, and succulence. Orange color and novel colors are important for consumers. And convenience is important. That's what drove the development of baby carrots, shown here to the right, because of their convenience. Nutritional quality is also important to consumers and to growers, but it's not something that's readily measured by growers or consumers. Let me go through a couple of traits that we've done some breeding of carrots for, tapping into that genetic diversity to improve the modern crop, both nematode resistance and breeding for carotene content. First, talking about nematode resistance breeding. The Mollidigani root knot nematodes are important pests of nematodes, pests of carrots in the large-scale growing regions of California. And in the early 90s, Phil Roberts and I discovered a single gene resistance for nematodes. And you see this carrot cultivar was grown in an infested field. There was some resistant carrots on the left, as you can tell, and of course the susceptible carrots on the right. Today, we found several additional sources of resistance. I didn't mention it, but initial resistance was from Brazil, a Brazilian carrot. In addition, we found resistance from carrot cultivars from Syria, carrots from China, carrots from Europe and carrots from Australia. So the nematode resistance has been derived from a wide range of sources, with the 0 to 1 score indicates a high level of nematode resistance. It's a relatively rare resistance. We have not found it commonly. But we have been able to take these multiple sources of resistance and then intercross them. Of course, the idea to pyramid these sources of resistance. And today we know there's roughly at least 5 regions of the genome associated resistance identified by Josh Parsons, who's a grad student in my lab. These 5 regions of the genome are on different chromosomes, which makes their incorporation somewhat easier as we're pyramiding these resistance genes together for nematode resistance carrots today. And we're using molecular markers as part of this selection. And so where we are today is we've identified resistance from diverse sources I mentioned, and we've got markers to select that resistance, and we've released 7 resistance sources of nematode resistance to the seed industry. Moving on to some activities in the genetic improvement of carotene content to improve carrot nutritional value. A little bit of background on carotenes and carrots. Carotenes are pigments occurring in all green leaves, and they're essential for photosynthesis. So that's why carotenes are in plants. And carotenes involve a large number of compounds, somewhere in the 600 range, that account for red, yellow, and orange colors. Only a few of these carotenes are vitamin A precursors, referred to as provitamin A carotenes. And in fact, all vitamin A in animals ultimately comes from provitamin A carotenes. A brief image showing pictures of, on the bottom of this slide, a picture of what the vitamin A molecule or retinol molecule looks like. Above that, you see beta carotene, which when we eat orange carrots is one of the 2 major compounds, alpha and beta carotene. This beta carotene gets cleaved in half, and each half becomes a molecule of retinol. Hence, we get this vitamin A when we eat orange carrots because of these provitamin A carotenoids in carrots. At the top of this picture is another carotenoid, lycopene. This is not a provitamin A carotenoid, but it is important for the color of red carrots and also for tomatoes and watermelons. Let's see here. So where are we at in terms of the need for vitamin A today? It's an essential nutrient. It's a vitamin for which there's a deficiency in much of the developing world, and accounts for hundreds of thousands of deficiency symptoms in children every year, with a couple 100,000 children dying from vitamin A deficiency annually. So where had we gone to in terms of improving carotene content, these orange pigments in carrots? This image shows some cultivars of carrots, and so what we did was intercross different orange carrots shown on the top left and the lower right of this slide, with the idea of combining genes from different sources to improve carotene content. The genes for carotene, there are 2 major genes that control the accumulation of carotenes in carrots. The Y gene, which accounts for the difference between white and yellow carrots, wild carrots and yellow carrots, shown on the bottom, and the Y2 gene accounting for the difference between yellow and orange carrots. We know these genes are not biosynthetic genes, but rather accumulation genes, with the Y gene involved in photomorphogenesis based on some recent work we did. And the Y2 gene, we haven't got it narrowed down completely, but down to about 10 candidates we're focusing on today as candidates for the Y and Y2 genes. So 2 major genes accounting for white versus yellow versus orange carrots. When we come to orange carrots, there are many genes accounting for the range in carotene content, which can go between 50 and 1,000 parts per million of alpha and beta carotene. So a wide range, and we've released several populations with high carotene content today based on intercrossing these diverse sources. Just to briefly talk about release of carrot germplasm as part of this series, this is all USDA ARS released germplasm, consequently it's freely available and open access. So where are we at in U.S. carrots today in terms of carotene content? Well, we know in the 1970s, carrots contained about 90 parts per million total carotenoids, alpha and beta carotene. These carrots had paler orange phloem in particular, and some yellow cores, whereas today carrots contain in the U.S. about 130 parts per million total carotenoids. So 1 square meter of high carotene carrots today provides enough vitamin A for several adults for a full year. Some images of what selection for carotene content looks like: a progenitor in the slices in the middle, and 2 different populations selected for high carotene. In the progenitor had 140 parts per million provitamin A carotenoids. The population on the left, 270, twice as much. Population on the right, 3 times as much provitamin A carotenoids with phenotypic recurrent selection. This was successful based on using diverse intercrosses to start with, as our other breeding we've done, and other breeders for that matter. And today we have molecular markers for several of those genes. And we have actually increased the carotene content quite substantially, comparing, I mentioned before, in the 1970s, 90 parts per million, compared to today's carrots, about 130 parts per million. At the same time, we focus on improving flavor, because pigments by themselves will not necessarily gather the interest of consumers as will better flavor. So I'll talk about flavor as we go along here too. But where we've come in improving the crop in terms of dollar value from 1975 to 2015, we've more than 50% increased the dollar value of carrots, partly due to more acreage, but also due to larger per acre value, and also increase the carotene content and per capita consumption, so that today carrots account for roughly 12% of the vitamin A in the U.S. diet. So a significant source, considering Americans typically only eat about 5 or 6 kilograms of carrots per year. This is a carrot breeding talk, but let me briefly step aside for a minute and talk about what can be done to make fruits and vegetables more generally better sources of vitamin A. One approach is simply to use varieties with higher vitamin A or provitamin A carotenoid content. I mentioned that there's this variation in carrots. You see an image here of different bananas where some of them have very high provitamin A content, and consequently those would be very good sources of vitamin A. In addition to using cultivars that are already available, we can proceed with a breeding approach like we've done with carrot. You can look at improving the productivity of crops that provide vitamin A, because in some cases the availability of the crop is limited. And so if we can grow crops that have a more nutrient-rich basis to them, this can translate to higher availability to consumers. And finally, if looking at ways to encourage consumers to increase intake, one approach is convenience. We know that baby carrots have increased carrot consumption to some extent. And also to look at improving flavor, because the consumer, whereas they're interested in nutritional value, it's actually flavor that is the driving force for repeat consumption. So that's where we are at in terms of a couple of traits for carrot breeding today. And the question then is, why bother improving carrots? You go to the grocery store, there's carrots there every day. What more could we possibly improve? I hope I've given you a couple of examples of where we've managed to improve some carrots for both growers and consumers for a couple of traits today. Who needs better carrots? I think anyone that's interested in consuming carrots could benefit from a better tasting and more nutritious carrots. To summarize what I've been talking about briefly, I mentioned carrots, a relatively recent crop, about 1,100 years as a root crop with great diversity. The initial development of hybrids began in the 1950s, and baby carrots in the 1990s. With the development of molecular tools that we have today, we anticipate this process of breeding will be accelerated. Carrot breeding for nutritional value has been successful, but there's still long ways we could go in improving nutritional content. Flavor improvement is critical to increase consumption. Like other crops, there are several diseases that pose a threat to carrot, and new ones no doubt will show up on the scene. And we've had some success in developing high-value carrot products in the U.S., but there's also much potential for additional products. Breeding carrots for low input and organic production is important today because the demand is rising, and already carrot is a fairly widely grown crop in the organic production area. And finally, we're very interested in expanding carrot production in more saline soils in warmer and drier climates of the world, because it seems that this is where the production regions of the world are going to be going. And so we're initiating some work in this abiotic stress breeding as well. So like this farm family in Wisconsin in the 1850s, it took this family to grow this table full of vegetables, and it takes a team for carrot improvement like any other breeding program. And that team has included some very important postdocs and support scientists, graduate students, and funding sources, several of which I've listed here. So I thank you for your time and attention in this presentation, and I would welcome any questions for any time remaining.
Okay, folks, feel free to put questions in Q&A. We have one last poll we're going to do on hybrids, a couple of hybrid questions to test your knowledge there and how well you were paying attention. So feel free to log in or log your results in the poll and submit those. While we're waiting, we did have a question in chat about someone said they'd always heard there was a connection to the Royal Dutch family with regards to the orange color in carrots. Is this true?
There's a connection in the sense that orange had been the color, sort of the national color of at least a part of the population of Holland. And so when orange carrots became available, they were very popular in Holland. It seems that Holland eventually was a place where orange carrots were grown, but the first orange carrots appear to be have originated from Southern Europe, and then eventually made their way to Holland. So that, yeah, the royal family of Holland was interested in them, and certainly have indicated and encouraged their consumption ever since.
Alrighty. So again, folks, if you have any questions, feel free to you can put those in Q&A or chat. So just a couple of observations I made, Phil, based on the information you gave. So there's a direct connection, and the orange color is directly tied to the amount of vitamin A that's in there. And that is to say that purple and these other exotic colors do not, or at least have lower levels of vitamin A?
Yeah, that's a great question. I didn't take the time to talk about that in detail. So the yellow, red, purple, and orange pigments all have some nutritional value. The only one that's a vitamin A precursor is the orange color. That's alpha and beta carotene. I showed you pictures of a purple carrot with an orange core. So that carrot has both those anthocyanin purple pigments as well as the orange pigments. So you really get both of them, and those purple pigments are great antioxidants. Red carrots have lycopene, which is another cancer-fighting compound, and yellow carrots have lutein, which is another important compound for health. But the only pigments that provide vitamin A are the orange pigments, alpha and beta carotene in orange carrots.
Okay, gotcha. So I'm going to end this poll, and I'll share the results now. And did folks get everything right there?
They did a great job. Yeah.
Okay.
Those of you that have hand-pollinated carrots, it's possible, but they're mighty small flowers, and you only get 2 seeds per flower. That's why cytoplasmic male sterility is essential for large-scale hybrid production. And in fact, it's the mitochondrial genome that is, like other cytoplasmic male sterilities, is the ultimate source of that male sterility that we use to make hybrid carrots today.
Okay. All right, we got a few questions. I'm going to stop sharing those results. Got a few questions coming in. So in chat, someone says, can Dr. Simon give a hypothesis as to why a wild carrot or any crop would have evolved a CMS trait?
That's a great question that we keep asking ourselves, and it's not just us. This is a widely asked question for those that utilize cytoplasmic male sterility. Obviously, a plant that is male sterile automatically will produce seed that's a hybrid seed, and so it allows for the expression of that hybrid vigor but then that plant very well may be male, the progeny may be male sterile, and that's a dead end. The whole origin of male sterility, at least from my reading of literature, doesn't have a particularly good explanation. So I will wait for future work to explain that better.
Okay. Surya in Q&A says, thank you very much for the nice presentation. You mentioned different parts of the world grow different types of carrots. Any particular reason, maybe flavor?
Yeah, flavor is a part of it. One interesting thing on flavor is that most of the carrots grown, especially in Asia, but also to a larger extent in Europe than the US, is that carrots grown in Asia and most of Europe are cooked, and often cooked quite a bit before they're consumed, whereas there's a large amount, around 85% of the US carrot crop is sold as a raw carrot, as baby carrots or whole carrots. Now, these are often cooked, and so anyway, that translates to some of the flavor selection that we have to focus on raw carrots more so than maybe the rest of the world. That doesn't necessarily have any association with the root shapes. Those are more traditional root shapes. The long, thin carrots are a bit more difficult to grow, and consequently, the cultivation necessary to get a marketable crop is more important than a shorter carrot. Otherwise, it's more just a matter of tradition.
Okay. Jody says, where have you traveled to collect carrot germ plasm?
That's been a very enjoyable part of my career. That along the way, I've been on about 10 collecting expeditions. We've collected in Central Asia, Kazakhstan, Uzbekistan, Kyrgyzstan. Did some collection in Syria, actually, when Syria was in better shape, Turkey, Greece, Morocco, Tunisia, France, Spain, and some collecting in the US. So I had an opportunity to collect in quite a few regions of the world, and it's really been a great experience.
Yeah, is there anywhere in the world that doesn't grow carrots? I mean, it sounds like
in warmer climates, it's a cool season crop, and there's not as much production in very warm climates. But every place I've been to has some carrot production. It's usually a high value crop and usually orange.
Sure. Okay, a couple more questions. Evan says, could you comment on the bitterness in carrots associated with metabolites and how breeding against these may have had effects on biotic stress resistance of elite cultivars?
Yeah, great question. So when I talked about flavor, I talked about sweetness. That's pretty well understood. The other flavor that's important for raw, fresh carrots is what I call harshness, which is a reflection of volatile terpenoid content. These are volatile terpenoids, some of the same compounds in something like pine needles. And in fact, some carrots taste pretty piney when you first harvest them out of the field, and it's due to those volatile terpenoids. A wide genetic range for those volatile terpenoid compounds. Many people refer to that flavor as being bitter. I refer to it as harsh because those volatile terpenoids are present at harvest, whereas there is the production of bitter compounds post-harvest in carrot roots when they're stored in cold storage with exposure to ethylene. So carrots stored in a fridge with apples or pears will get bitter. And this is a similar flavor to the harshness, but it's strictly a post-harvest flavor. Also, it's a flavor that you often can remove by peeling carrots, whereas this harshness I mentioned, it goes through and through the carrot root. So this bitterness is caused by some phenolic compounds, and again, it's associated with exposure to ethylene. It's thought that these bitter compounds may impart some level of post-harvest disease resistance, although that's not been clearly shown. In fact, the reason that volatile terpenoids or these bitter compounds are important in carrots are not particularly known. In fact, carrots that aren't harsh and aren't bitter don't necessarily have more disease susceptibility in their field or post-harvest. So we don't quite know what those compounds are doing. We do know that they have a huge impact on flavor.
and there's no way to get rid of that. I actually had the experience. It's sort of fortuitous that I'm on this webinar, and I had the experience of growing some carrots last year. And I think I complained to my dad, who's been gardening much longer than me, that, man, they bitter. They just really didn't taste good at all. And he asked me how long I left them in the ground. I said, well, they might have been there 3, 4 months. And he said, well, that was probably your problem. You let them grow too long.
Yeah.
And there's no way to get rid of that bitterness, though, sort of once they've developed to that point.
Not really, no. I think your best bet is find a cultivar that doesn't accumulate these terpenoids, and you're better off.
Okay, good. Okay, we had a question in Q&A from John. He says, in many crops, there is an indirect correlation between productivity and various quality traits. Are there any of these issues in carrots?
Yeah, that's an interesting question. We see, I didn't talk much about white or yellow carrots. There are white cultivated carrots. You can see some in this slide here, as well as white wild carrots. And white and yellow carrots tend to produce larger roots. So these yellow carrots have pigments. So obviously it doesn't translate to lack of pigments, only our larger roots, but there may be a little bit of a productivity cost with the production of the pigments. To be honest, as a marketable crop, we look for smaller carrots as baby carrots, for instance. These are thin carrots that are not necessarily growing for a long time. So there probably is a bit of a trade-off. We haven't actually quantified that, but not too large, and primarily for those colors other than white and yellow.
Yeah. Well, I'm Googling carrot seed as we speak, just because this has sort of renewed my interest in maybe growing some different varieties of carrots this spring. Okay, so we got about 8 minutes left, so still time for questions. Evan followed up and says, do carrots left to, I may be saying this wrong, senescence or
senescence, maybe?
Okay, senescence at the end of their first year seem to accumulate bitter compounds as a result of ethylene production in senescing leaves?
Yeah, that's a good question. It has been noted that carrots left in the field, as you noted, Mark, can be somewhat, apparently Evans noted too, that they can be rather strong flavored. And so a couple of, well, first of all, there's no clear answer on it. From what we've looked at, these volatile terpenoids will keep accumulating, but tend to level off as the carrot root gets sized up as harvestable size. One change that does happen as a carrot crop is left in the field later in the season is that it can be that some of the sugars that have accumulated during the more optimal growing season might be utilized in metabolism and consequently may be less sweetened. So some of that harsh flavor may show up a little more pronounced manner if the carrots have been in the field longer. But short answer is we haven't really done much extensive studies on this question. But it's been a recurring observation. Haven't gotten to it studying it yet, though.
Okay. Okay, we'll take another minute. No questions at this time. We'll take another minute and take time for questions. Chris, do you want to, while we've got a silent moment here, you want to let folks know about the next webinar in our series?
Absolutely. And so as part of this series, we've been focusing on a variety of topics, including intellectual property. We had a special topics webinar on high throughput phenotyping and unmanned aerial drones. And we're very grateful today for Dr. Simon talking about carrot breeding and all the good information there. And so in 2 weeks, we'll be back here with Dr. Jim Myers from Oregon State University. And he's going to continue with the theme in intellectual property. He's going to be discussing trends in intellectual property in protection in plant breeding. So he's going to offer an additional perspective to what you've heard and a lot of information that will tie in to the other topics. So we hope you can join us in 2 weeks.
All righty, I'm not seeing any additional questions, so I think we'll, well, there's one just popped into Q&A. Yes, so someone asked, is this presentation going to be available online? It will be, and Chris, if you got the Learn link handy, yes, again, if you'll post that in, yep, most of you probably joined us through learn.extension.org on that same page. Within a couple of days, we're going to update the link. There'll be a link to the recording there, and then we'll also try and get, if Phil is agreeable, we'll get his slides and we'll convert those to a PDF maybe, and those will be available in the event materials section on that same Learn site.
I'll get those slides to you tonight, Chris.
Okay. All right, Phil, any closing thoughts or comments?
I'll make one comment. Wild carrot in North America probably came from Europe. It was not a native crop, but probably came as a weed with our first European settlers based on some work we've done. So it's a widespread phenomenon throughout North and much of South America today, but it originally probably came in on the shoelaces or pants cuffs of our European ancestors.
Great. Okay. All right, Phil. Thank you so much. This was a great webinar, and everyone have a great growing season. Now is the time, I guess, to be putting seed in the ground. So yeah, do that. And as Chris mentioned, we'll be back here in another couple of weeks.
Thank you.
Thanks, everyone. Bye-bye.